Welcome and thank you for standing by. For the duration of today's conference, all parties will have an open line. If you are not speaking, either utilize the mute feature on your phone or you may press *6 to mute and unmute your line. I'd like to inform all parties that today's conference is being recorded. If you have any objections, you may disconnect at this time. I would now like to turn the conference over to Diane St. Germain. Ms. St. Germain, you may begin.
Thank you. Hello, I'm Diane St. Germain with the National Cancer Institute. On behalf of the International Society for Quality of Life Research and the National Cancer Institute, I would like to welcome you to the webinar series titled Best Practices for Integrating PROs in Oncology Trials. The webinar series is comprised of 6 webinars, each approximately 45 minutes in length. The webinars are designed to be viewed independently to meet your individual learning needs, or the series can be viewed sequentially in its entirety. Today's webinar is the second in the series and is titled How to Select the Appropriate PRO Measure, presented by Dr. Bryce Reeve and Dr. Ethan Bosch. I will now turn it over to them.
Thank you very much, Diane. And so as a quick introductions, my name is Bryce Reeve, and I'm an associate professor in health policy and management at the University of North Carolina. I'm also a member of the Lineberger Comprehensive Cancer Center. My background training is in psychometrics, and my research is focused on both the development of patient-reported outcome measures and also their use in clinical research and clinical practice. And I'll now allow my good friend and colleague Ethan to introduce himself.
Hi, hello. My name is Ethan Bosch, and I'm an oncologist and a health services researcher, also at the University of North Carolina, and also a member of the Lineberger Comprehensive Cancer Center. And my own research focuses on the development of patient-reported outcome measures for use in various oncology contexts, as well as the integration of those measures into various healthcare processes.
Thank you, Ethan. And so, as on the title slide, Ethan and I will be talking today about how to select the appropriate patient-reported outcome measure for use in the oncology clinical trial. And this is the second in the series for the webinars. What I'd like to point out with this next slide here is, in thinking about what we're trying to measure, which are patient experiences and patient perspectives, what I like about this quote from this German physicist, Werner Heisenberg, it says, what we observe is not nature itself, but nature exposed to our method of questioning. And what I like about this quote is it recognizes that, number one, is we believe the patient is the gold standard. However, we recognize that through our means of collecting the patient experiences and perceptions through a standard questionnaire, it's going to have some error or flaws with it. We're never going to get a true perfect measure of what the patient's experience it is, but it is our job and our task to try to continue to find better ways to develop better measures that can measure both the impact of cancer and treatment on patient lives. What I also like about the slides, it recognizes is that people within our country and internationally have different perspectives and experiences. And this is informed and shaped by perhaps their race, ethnicity, the language they speak, their age, the type of cancer they have, and treatment. And again, we have to consider all these issues in thinking about what is the right measure for our particular population. So today, Ethan and I have this outline set up to talk with you today to go through these key points there. At first, I'll briefly talk about identifying what is the right PRO that addresses a research question. I'll talk briefly about what are the key attributes of a quality patient-reported measure. And then after talking about what are the attributes and qualities we're looking for in a measure, Ethan and I will talk about the different type of measures that are out there. And I'll talk about the differences between both the generic and the disease-specific patient-reported measure. Ethan will talk about two key patient-reported outcome measures that are supported by the National Institutes of Health. That includes both the PROMIS and the PRO-CTCA measures. And then Ethan and I will tackle some key questions that are commonly asked among investigators about the different types of PRO measures for different types of research studies. And then we'll end up with some final recommendations and recommend some key references. So let us start with the first part of our outline, which is identifying the right PRO that addresses your research question. So stepping back, we have to recognize for clinical research that patient-reported data can play multiple roles in a clinical research study. Traditionally, what we'll be focused on most of today is that the patient-reported data is used as an outcome measure, whether it could be a primary outcome in a symptom management trial where the study is trying to reduce levels of pain, or the patient-reported data can be used as a secondary endpoint where perhaps the primary endpoint is survival and the secondary endpoint may be which intervention results in perhaps less fatigue or less nausea. In addition, if there are no pre-specified hypotheses, the patient-reported data can be used in exploratory endpoints that helps provide sort of a general profile of the health-related quality of life of those individuals in the different intervention arms. Another key aspect and use of patient-reported data is using patient-reported data to inform adverse event reporting. We think this is a key new field of development, and later in our presentation you'll hear Dr. Ethan Bash talk about his work in developing the patient-reported outcomes version of the CPCAE that's used in oncology clinical trials. In addition, patient-reported data could be used as an eligibility screener in a clinical research study. And so if you were looking at the effective intervention for patients that are experiencing high fatigue or high pain, there might be a patient questionnaire or a patient item that pre-screens a patient to make sure they are experiencing moderate to severe levels of fatigue or pain, whatever happens to be the eligibility for that particular study. In addition, patient-reported data can be used to help to serve as perhaps a mediator in a research study. In this instance here, for example, there might be interest in how a particular drug or non-pharmacological behavioral intervention might help a person who suffers from migraines to improve their daily living, be able to return to work or carry on their home activities. In this model here, while the primary outcome is interest is that person's return to work or their daily activities there, however, the intervention itself might, again, the medication might actually have impact on the symptoms of migraine, and then once those symptoms are relieved, then that person is able to return to work. So the domain of migraine symptoms is the mediator study that is in between both intervention and the final outcome. In addition, the patient-reported data could serve as a moderator, where the effectiveness of an intervention is moderated or depends on the level of the moderator variables. So in this study here, we might be looking at the intervention like meditation and looking at the impact of meditation on depression. Well, it may be that meditation works well for those who are experiencing high levels of stress. And then when their stress is relieved through meditation, they might have reduced depression. However, for another group that experiences no stress, the meditation might have no impact. So there, the variable of stress and the level of stress is used as a moderator study. So what this slide presents to you is we need to think about how the patient-reported data plays the role in the study, and depending on its role, might require different types of patient-reported measures, whether they might be a single-item measure for an eligibility screener, multiple-item measures for an outcome, or different types of measures based on whether it's, again, a mediator or moderator variable in the particular study. And so now that we understand the key roles of PRO, one of the most important aspects in our sort of the design of a study here is before we start selecting an instrument, we need to help understand is what is the outcomes we want to measure in our research study. Now, this particular topic is covered in our first webinar led by David Sella, so I'll just briefly cover some of the key points. The most important thing, again, before we choose an instrument is we need to know what is the outcomes we want to measure, and this depends on the goals or aims of the particular research study. So we need to know the context of the clinical trial. What are the research questions? what are the hypotheses, and how do they relate to patient-reported data. This includes both an understanding of the characteristics of the population, their age, disease burden, race, ethnicity, etc., and then most importantly is how this intervention will help, will change particular outcomes, whether they might be fatigue, pain, stress. So you need to have an understanding of the context intervention to help before you start selecting which PRO measure to use. In helping to inform which outcomes to measure, it's important to involve key stakeholders, which includes both content experts, researchers, clinicians, and especially patients. It's important to have patients involved to help determine what are the most priority outcomes to measure in a clinical study. And as noted in the third bullet below is these steps need to be done before you choose a particular PRO measure. So once the PRO domains have been identified, it's next time to turn to, how to select the most appropriate measure. And so that's what we'll turn to now in our outline, which is what are the attributes of a high-quality measure. And so I'm going to list 8 attributes that are identified by the Scientific Advisory Committee of the Medical Outcomes Trust. And these are key attributes that you would look for to help you identify what's the appropriate measure for your particular research study. And so in searching for a measure, often you'll find information about these attributes in a key psychometric publication or in some other document that supports the validity and reliability of that particular measure. So let's run down through these 8 attributes, as all of them are key features you want to look for when selecting your PRO measure. The first one is the conceptual and measurement model. The conceptual measurement model provides the rationale for and description of the outcome or PRO concept and the populations that this particular instrument is intended to assess in the relationship among these concepts. And so it's important is for any particular measure, it needs to have a conceptual model which helps drive how the items and the attributes that are measured in that particular instrument. Without that conceptual model, there's no theoretical perspective for what type of content should be measured with that particular instrument. And then once that conceptual model is identified, then that helps derive what actual items appear in that particular instrument. So let me use an example in the area of, in the area of measuring the concept of fatigue or tiredness. And so if I were developing Bryce's new measure of fatigue or tiredness there, I would want to start with a conceptual model which helped me to understand what aspects of fatigue or tiredness I want to measure in my clinical study, in my patient-reported outcome measure. And so this conceptual model, which is from the article of Bartzovic et al., listed at the bottom of the slide here, notes that the experience of fatigue or tiredness includes aspects of both the subjective experience, which is the severity of fatigue and tiredness, how worn out a person is, but also includes aspects of how fatigue impacts their physical functioning, cognitive functioning, emotional functioning, social functioning. Then with this conceptual model, this helps me to understand what kind of items to include in my particular measure of fatigue. And so what I would expect to see in the instrument and developing the instrument there is that there should be specific items that map to each of those particular 5 attributes of fatigue or tiredness. And in this particular example, which is only an example here, is you'll see there are 3 items that measure each of the 5 attributes of overall fatigue. The second concept or attribute of a quality measure you want to look for is in the area of reliability or precision of the instrument. And reliability is the extent to which an instrument can distinguish between one population or one group or another group where there are true systematic differences, even after adjusting or accounting for the presence of measurement error in that particular instrument. And when thinking about reliability, let's think about when we look at an observed score in a patient-reported outcome instrument. Any score we get on any patient-reported outcome measure consists of two attributes. One is the actual true score of a person's, the true level of a person's fatigue level, and some aspect of measurement error. When you think we're trying to measure someone's true score, if you can imagine there's a whole population of items that could be included in a PRO measure. And so for the area of fatigue, there could be hundreds of thousands of different items you can imagine that measure a person's fatigue level. However, we recognize that we can't obviously administer hundreds or thousands of questions on fatigue to a particular patient. And so we choose a subset of items that we think will characterize that particular domain. And those particular subset of items will produce an observed score, which will be a reflection of the true score, but will also have measurement error because you were unable to administer every single item, as well as other aspects of error that will be associated with measuring someone's true fatigue levels. Reliability is basically looking at what proportion of our observed scores is related to a person's true score. And there are at least two different types of reliability we're interested in when looking at the reliability of the patient-reported measure we're trying to choose for a particular study. The first type of reliability is called internal consistency. And this basically looks at to what extent is the instrument free from measurement error. And this can be assessed in a cross-sectional study. And it typically, for internal consistency, is interested in looking at the association among the items that make up a particular measure. So if you have 5 items that measure fatigue, internal consistency basically looks at the association or correlation among those 5 items. There are at least 2 ways you can measure internal consistency. One is using Cronbach's coefficient alpha, which is bound between 0 and 1. And another way is using item response theory modeling and looking at information functions, which is beyond the scope of this, but something worth looking into to looking at the internal consistency of a measure. Another way of looking at reliability is what's called test-retest reliability of the instrument. And so this assessment of reliability is when you administer the questionnaire, again of fatigue, and you measure at 2 time points, maybe at day 1 and then the next day, when you expect the group or population to be stable or not changing. And so the extent that they provide similar scores on both assessments is a measure of the test-retest reliability. The association we typically look at to look at test-retest reliability is interclass correlation coefficients. So again, for internal consistency, we can get that information from a cross-sectional 1 time point study. For test-retest, we need at least 2 time points to assess the reliability. And what you're looking for in terms of a standard here is ideally for the reliability, again, is bound between 0 and 1, where 1 represents perfect reliability and 0 represents no reliability and 100% error, is ideally for clinical research where you're doing group-level comparisons, you want your reliability at least to be above 0.7. The next attribute we're looking for in a measure is the attribute of validity. And what the attribute of validity basically is looking at is to what extent is our patient-reported measure a reflection or a measure of what it's supposed to measure. Now, there are different types of validity that are important to consider when selecting an appropriate patient-reported outcome measure. One of those attributes, which is well discussed, is the attribute of content validity, which looks at the extent that the instrument or patient-reported outcome measure includes items that measure the attributes that make up the measured construct. And this relates to the concept I noted in my earlier discussion related to the conceptual model for a particular measure here. And so what we see in this slide here is that conceptual and measurement model for the concept of fatigue or tiredness. And so to evaluate content validity of our Bryce's new fatigue measure here, we expect it to include all 5 attributes, items that measure all 5 attributes of this concept of fatigue. Are there items that measure the subjective experience? Are there items that measure impact on physical functioning, cognitive function, emotional function, social function? So what we're looking for in content validity is more of a qualitative assessment, and this can be done both in focus groups or in-depth interviews, is making sure through content expert eyes we have items, again, that map to all the attributes that relate to conceptual model of the PRO measure. Another aspect of content validity here discussed is this issue of face validity, which is when you look at items that make up that particular patient report outcome, items or questions there, it should make sense that when you read those items, when a patient looks at the items, they can describe to you that item measures things that are consistent with what you're trying to measure. So, for example, if there's an item on my new fatigue measure that says, in this example at the bottom here, in the past 7 days, to what degree does your fatigue or tiredness interfere with your physical functioning? And you can see the response options from not at all to very much there. Well, a patient could probably tell you that, well, this question is talking about my fatigue or how tired I am, and this interferes with how my ability to to go walking or walking upstairs. And I think in that way, that's an assessment of face validity. It's a very sort of simple way to assess it, and it's typically done in in-depth interviews with patients where they're able to describe what they think that item is measuring, and as they describe it, it relates to what you as a researcher are trying to measure with that particular PRO measure. The second type of patient validity that is less discussed or talked about is criterion-related validity. And criterion-related validity measures the extent to which a measure agrees with some external standard, which is typically a gold standard measure. And so the reason why you don't see criterion-related validity discussed too much in the psychometric validation of the instrument is because often in the area of patient-reported outcomes where we're measuring subjective experiences such as fatigue or depression or anxiety, we often lack a gold standard to compare our new measure to. One exception, which is a good example of looking at criterion-related validity, and one aspect called concurrent validity, is if I developed a new measure of physical activity, and my questionnaire asks, to what extent can you use a hammer to drive a nail in a piece of wood, or are you able to walk up and down a flight of stairs there, is after completing the questionnaire, we can have the patient actually go and complete the exact same exercise. So if they say they can hammer a nail, you can give them a hammer, give them a nail, and see if they can do it. If they say they can walk up and down a flight of stairs, you can actually have them do it. So there's an example where you can actually assess criterion-related validity called concurrent validity and actually have a person report on the PRO measure and then perform that particular exercise. However, for many of the other types of domains we measure, we just don't have that type of gold standard measure. The third type of validity, which is extremely important to assess for any particular patient-reported outcome measure and equally important as construct validity, is the notion of construct validity, which is, does our measure behave in a way that's consistent with any theoretical hypotheses you may have about how that measure should behave, and how the instrument represents how well scores on the instrument are indicative of the theoretical construct. And there are different types of construct validity you'll see mentioned in the published literature. One type of construct validity is structural validity, and this looks at the extent to which the items measure a single or multiple factors in your scale. So for fatigue, what we expect that there's one single domain of fatigue. When we do a factor analysis in terms of structural validity, we expect only the factor analysis to identify one domain. However, if I had another patient-reported measure that was looking at an area of emotional distress, which includes anxiety, depression, and anger, I would expect under the notion of structural validity that that if I did a factor analysis, I would expect to see three factors emerge: anxiety, depression, and anger, as to be both distinct but related concepts of emotional distress. Another type of construct validity is convergent validity, which is looking at how my new measure is related to some existing measure that's used in clinical research. So again, if I had Bryce's new fatigue measure, to look at convergent validity, I could compare my new fatigue measure versus an existing measure like the FACT fatigue scale and see the extent that they should correlate together, which is what you would expect. Discriminant validity, which is also referred to as divergent validity, means that how well your new measure should be less related to concepts that have less relationship with what you're trying to measure. So while convergent validity looks at how my new fatigue measure is associated with other fatigue measures. I can compare my new fatigue measure versus other domains like nausea or depression, where I would expect there might be some correlation, but there also might be not as strong as compared with another measure that's measuring fatigue. And then the last form of construct validity is known groups validity, where if you expect one group to have higher scores than another group on your measure, you can actually empirically test that. So, for example, for fatigue, I could compare a group of cancer patients that are on chemotherapy versus a group of cancer patients off therapy, and I would expect those on chemotherapy to report higher levels of fatigue. So, moving on to the next slide. And Pam, if I could have you move to the next slide for me, please. I've lost my cursor. There you go. Thank you. I found it again. So the next aspect of an attribute of quality instrument is the responsiveness of the PRO measure. Responsiveness is actually one form of validity, but it's pulled out by itself there, and it measures the ability of an instrument to detect changes over time. And so what you're looking for in that instrument there is some type of evidence that that measure used in the longitudinal research study is responsive to an intervention's effectiveness. So, for example, for the domain of fatigue, I could measure a group of cancer patients at baseline before chemotherapy, and then measure the fatigue within treatment, and I would expect my new fatigue measure to show an increase in fatigue when they're on chemotherapy. And again, that's a measure of responsiveness, which is another key attribute of a particular measure. The next attribute of interest is interpretability. And so what is very important about this attribute of any PRO measure is you need to understand what the scores from this measure means. And if the researcher is unable to interpret what the score means, then it's not a very valuable instrument. So, for example, if you had a group mean or individual mean of 43 by itself, you don't know what that means. And so we need to find better ways, the instrument needs to find better ways to communicate to the researcher what this score means. Is 43 mean high fatigue, low fatigue? How do other cancer patients score in this measure? And so when looking at a particular PRO measure, there should be a good description for what do high scores on the measure represent. Do high scores represent high fatigue, and low scores represent low fatigue? The developer should provide you what the range of scores are. Do the scores range from 0 to 100, 0 to 50, 0 to 1,000? It also might be informative, and some measures is what's called reference norms. And so it may be for my particular fatigue measure for the general population of either the U.S. or cancer patients, the mean score on average might be 50 with standard deviation of 10. And that's helpful to understand how your group or your patient differentiates from some specific reference norm, again, whether it's the general population or population of cancer patients. What's important is when you see changes in the score across time or across groups there, is how do you know if those changes are meaningful? And so you might see discussions of what's called clinically significant change scores or minimally important difference scores, and whether a 5-point or 10-point change are important both clinically to the patient and to the researcher. That would help, again, inform how interpretable those change scores over time. So all these aspects are important attributes to consider when selecting a PRO measure. The next attribute you want to consider when selecting a PRO measure is, most importantly, is respondent burden. Obviously, in oncology practice, in oncology research, we have very sick, perhaps older populations who don't want to be burdened by very long questionnaires. So it's very important to consider when selecting a PRO measure is try to select one that has the least burden, the less number of items. However, recognizing is that you don't want to trade off reliability precision, because as you get to less and less items, you are you're increasing your measurement error. In addition, you need to also be aware of administrative burden and consider what are demands on the administrators to administer a shorter versus longer questionnaire, and whether it can be done on a computer versus paper, and how they're going to collect and score that particular instrument. The next attribute to consider when selecting a PRO measure is to what extent does that measure has been both used and validated for use in different types of administrative modes. And so does the instrument allow a person to self-report their fatigue or pain levels, or is the interviewer has to administer the instrument? Has the instrument been validated for use on paper format, on computers, or has the instrument been used for telephone surveys, or validated on telephone surveys? Telephone surveys actually can be an important component of a research study, because it may help you access patients who may be either too illiterate and unable to read a particular form, or maybe a hard-to-reach population where people prefer answering over phone versus completing on a computer questionnaire. So again, you're looking for a measure that not only has been used in different types of administrative modes, but also validated in those different types of modes as well. And then the last attribute to consider when selecting a measure is reflecting that the U.S. population, or internationally, we have a multi-ethnic, multiracial population. It's important when looking at the validity evidence is the extent that the validation of the instrument has been done in people of different race and cultures, as well as make sure that the instrument is available in languages that reflect your target population. So they're in English, Spanish, French, and other versions have been developed for that measure, and have they been validated in each of those particular translated forms. Those are the 8 attributes and information we look for to help you inform the selection of a particular PRO measure. And so what I'd like to turn to is talking about the different type of measures that are available out there. And one key difference are the differences between generic instruments and disease-specific measures. Generic instruments are patient-reported measures that have been developed for use in patients of different chronic disease settings or in the general population. And examples of generic instruments include the Medical Outcomes Trust Short Form 36, or SF36, which there's a version 1 and version 2 available, which is commonly used in clinical research studies and observational studies and population studies. The PROMIS measures, which you'll hear Dr. Bash talk about in a moment. For pediatrics, there's the PED-QOL. And there are also symptom measures, the Memorial Symptom Assessment Scale, Rotterdam Symptom Checklist, and there are many other generic measures which I'm not listing here. Now, the value of a generic instrument is that it can measure such health-related quality of life domains, such as fatigue and physical functioning there, and then allow you to compare your group versus some general population. And as an example, this is a study conducted by Dr. Ron Hayes looking at the burden of HIV in populations and comparing HIV populations with other populations with chronic diseases. And so you'll notice, and he used the SF36, and what you'll notice in the bottom 3 rows of this graph here are patients with HIV at different stages, from asymptomatic, symptomatic, and AIDS. And then he wants to compare their mental health, which is the red bar, and physical health, which is the yellow bar, versus other disease populations. And since this is the SF36 has been normed, where if you look at the general population row, which is the 4th from the bottom there, is to have a mean of 50 and standard deviation of 10 in the general population. And so because Dr. Hayes used the SF36, he can compare and see how both the mental and physical aspects of different stages of HIV compares to other chronic diseases such as multiple sclerosis, diabetes, depression, epilepsy, and so on. However, the downside of generic measures is they might not capture symptoms or quality domains that are specific to cancer patients. So most of these measures, like the SF-36, do not include questions about nausea and vomiting or on symptoms related to neuropathy. And so because of that, we have disease-specific measures that can include content or core life domains that are relevant to the particular domain of interest. And in cancer research, some of the major questionnaires that are used are examples of the ERTC, the FACT measures, the LASA measures, and the MEDASI measures. And those measures include symptoms and functional impairments that are specific to cancer, and those instruments also include specific modules that are relevant to particular either disease, type of cancer like breast cancer or colorectal cancer, or to particular types of interventions like chemotherapy trials or radiation trials. So that's the differentiation between generic and disease-specific measures. And now I'd like to turn over to my good friend and colleague, Dr. Bash, who will continue with our discussion, particularly of the PROMIS and PRO-CTCAE measures.
Thanks so much, Bryce. So I'll start by briefly describing the Patient-Reported Outcomes Measurement Information System, or PROMIS, which, as Bryce noted, has been developed with support of the NIH. And you can see the web URL on this slide for additional information. This is a screenshot of the PROMIS website, which has extensive resources available related to PROMIS, including the questionnaires, background papers, general information, and access to Assessment Center, which is a questionnaire administration platform. The goal of PROMIS is to improve the assessment of self-reported symptoms and health-related quality of life domains, crossing multiple different chronic conditions. And multiple different domains are included in PROMIS. And one of the advantages of using a system like PROMIS is that one can evaluate these various symptoms either individually or can assess multiple different symptoms. Moreover, because of the nature of how PROMIS has been developed, various approaches to questionnaires can be used, including shorter forms, longer forms, or using computerized adaptive testing, depending upon the particular needs or context in an implementation, either in a trial or in clinical care. And you can see a number of the domains listed on this slide. In addition, PROMIS has been developed both in adult and in pediatric populations. As Bryce also alluded to, advanced qualitative and quantitative statistical methods have been used to develop PROMIS, as well as a broad range of stakeholders, including patients and caregivers, in order to assure that the measures are high quality, but also salient to the populations in which they may be administered. In addition, as I mentioned, there's quite a bit of flexibility in using PROMIS because one can tailor both the specific symptoms and the level of detail of the questionnaire given the context of use. The PRO-CTCAE, or the Patient-Reported Outcomes version of the Common Terminology Criteria for Adverse Events, is another system that has been developed largely with National Cancer Institute support, and there is both an adult version as well as a pediatric version in development, and you can see the web URLs listed here. The PRO-CTCAE, as I'll describe in a moment, is a newer measurement system which is available through material transfer agreements with the NCI currently. So briefly, the PRO-CTCAE is a measurement system for patient self-reporting of toxicities or symptomatic adverse events, and it maps to the CTCAE, and this represents an effort to improve the accuracy of the grading of symptomatic adverse events in oncology trials. The adult PRO-CTCAE includes a library of items which encompasses 78 specific symptomatic adverse events. These range everywhere from sensory neuropathy to pain, to nausea, to bed sores, to certain types of transfusion reactions. Essentially, any of the domains within the CTCAE, which is the system for professional ratings of adverse events, that are amenable to patient report. The pediatric Pro-CTCAE is currently undergoing evaluation and is designed to measure approximately 63 symptomatic adverse events, which have been found to be amenable to reporting by children and potentially by caregivers. An electronic version of the Pro-CTCAE is hosted at the National Cancer Institute. So I'm now just briefly going to turn back to Bryce, who will describe this slide, and then he'll turn it back to me to finish up the presentation.
Thank you. So oftentimes we're asked a series of questions about how to select appropriate PRO measure, and so Ethan and I have listed a few of the key questions we received here, and one of them is, what are the advantages and limitations associated with using a single versus a multi-item PRO measure? So, for example, for the domain of depression, do I just need one single question, which asks, to what extent has you been sad, and have you felt sad in the past 7 days? Or should I use a multi-item scale where there might be 10 items that capture different attributes of depression or some other domain you're measuring? And so, the choice between a single or multi-item PRO measure, there are both advantages and trade-offs with either choice. And we have seen both single and multi-item scales used both in clinical research and clinical practice. So one of the advantages of a single item measure, obviously, is its less respondent burden. It's very easy for a person to complete one question, especially if you're going to have assessing patients' symptoms or functional status on a daily or weekly basis there, where they don't want to complete very long questionnaires on a frequent basis. And so, again, less respondent burden. Also, we find when we compare empirically the scores from single items versus scores from a multi-item scale, is there are typically moderate strong associations with of the single scale with the multi-item scale. However, there are some setbacks with using a single item measure, and these are issues to consider. One is, as I discussed earlier, for content validity. Again, content validity is, does your instrument measure all the different attributes of a particular PRO measure? And for fatigue, we noted that fatigue includes both the fatigue and tiredness experience, but also its impact on physical functioning, mental, social functioning, and cognitive functioning there. And when you have a single item measure, you can't ask about all those various attributes. So again, there may be some concerns about content validity of a single item measure. Whereas for a multi-item scale, it can include items that measure each attribute related to content validity. Another issue or an important thing to consider for a single item measure is the issue of reliability. As I noted earlier, is with a single item, there'll be a larger measurement error, and when there's larger measurement error, there's less reliability in the scale because you're only getting responses to one item, which might be in a 5-point scale, and so you'll be limited in being able to distinguish between groups that truly differ on your study. Whereas with the multi-item scale, it helps enhance or improve the reliability of the scale because of decreased measurement error. And then finally, with a single item measure, you're limited in the type of psychometric analyses you can do. With a single item measure, you can't look at internal consistency because there's no other item to compare to. You're limited to test, retest reliability. Whereas with multiple item scale, you can look at internal consistency, but also conduct such things as factor analysis, item response, and other things to look at the properties of the instrument itself. So those are trade-offs and issues to consider when selecting the measure.
Thanks so much, Bryce. So what to do if no patient-reported outcome measure exists for a particular concept of interest? The options are to adapt an existing measure or potentially to create a new measure. What are the advantages of each? Well, for an existing measure, very small changes in item wording may be acceptable without requiring extensive revalidation or repeat measurement of the properties of that tool. However, the more modifications that are made to a particular questionnaire, the less confidence one can have that it will perform the way that is expected based on prior development work. And the more likely one will need to conduct further qualitative or quantitative assessments of the properties of that tool. And therefore, if the needs of a particular context are beyond minor changes, there may be a need to create a new PRO measure. For creating a new PRO measure, one needs to follow best practices, and there are multiple documents available which outline current thinking about best practices for the development and testing of novel measures. And this is beyond the scope of the talk today, except to say that one should work with PRO development experts who are aware of this literature or these standards in order to develop a metric which is in keeping with those standards. And notably, the process can take variable amounts of time, depending again on the specifics of the metric, but one should be prepared for fairly rigorous and occasionally extensive qualitative and quantitative assessments. So what if the PRO instrument is measuring the right domain, but has been validated in a population different from the target population? For example, one is interested in measuring pain, and the pain instrument has been developed in the context of patients with osteoarthritis, and one is interested in measuring pain related to metastatic disease to the bone. Well, to the extent to which one can justify that the original validation sample is similar to the target population, the less additional validation evidence one will need to support the use of the PRO measure in that population. That said, it is often beneficial to do some qualitative work at least in such a setting. The greater the differences between the populations, such as in the example that I outlined, the general suggestion is to do some additional work, and the amount of work will really depend upon the needs as well as the potential burden and costs. And some of the considerations are listed below. One can obtain feedback from key stakeholders, including measure developers, clinicians, researchers, and patient advocates. One can move to qualitative work through in-depth interviews or focus groups towards concept elicitation and cognitive developing. And one could move further if the context of use is quite different from that which was originally tested, to conduct a pilot study to evaluate the measurement properties of that tool, including the reliability and the validity. And this can be nested into an ongoing clinical study, for example. In the setting of clinical research, if one is interested in ultimately using the tool in a phase 3 trial, and there is ongoing phase 2 work, one could embed the tool within the phase 2 study in order to look at its properties within the population of interest, and then one will have a greater understanding of how that tool performs before designing the use of that PRO in phase 3. So in summary, one should select a measure that best matches a patient-reported domain, has high adherence to existing psychometric standards, has evidence for the validity of the measure in the target population, is meaningful and relevant to the target population, including certain segments of that population that may have different responses to that questionnaire depending on their background, and which is feasible in a given research setting. Listed here are two references that may be helpful in further exploring some of the concepts that were discussed today. On behalf of Dr. Bryce Reeve and myself, Dr. Ethan Bash, I'd like to thank you again for your attention today, and we wish you luck in your future research endeavors. Thank you.
That concludes today's conference. You may disconnect at this time.
Okay, this is Pam. Thank you.
Thank you, Pam. Did you want to go back into a post-conference, or?
No, I don't. I think we'll just touch base later on.
Okay, very good. Thanks so much and have a great day.
Okay, thanks everyone.
Okay. Goodbye now.
Bye-bye.